Hello and welcome back. We're starting part 3 of the chapter on the heart. We left off with ventricular tachycardias and we're moving on into the mechanical aspects of the heart. Basically, the heart is a great big muscle. It contracts once it is activated with electrical activity. The chambers of the heart contract in order. The atria have to contract first, and then they end up filling the ventricles, and then the ventricles contract. What comes out of the ventricles is what's known as the stroke volume, and that's going to go into the aorta for the systemic side of things from the left side of the heart, and to the pulmonary artery from the right side of the heart. The stroke volume, how much comes out of the ventricles, is going to be the difference between the full volume, the end diastolic volume, that's the total volume before the heart contracts, and then take away from that whatever's left over. That's going to be the end systolic volume. So the leftover volume after contraction. There's some stuff still left in the ventricle. And so you have the total volume minus the stuff that got left behind. That's the stroke volume, the stuff that actually leaves the heart. And then this concept of cardiac output, what's coming out of the heart in a minute, is the heart rate. How many times did the heart pump, and how much came out each time it pumped, that stroke volume. There's a lot of information on this slide. We're going to walk through it slowly. All right, so cardiac muscle, it's a muscle. And like all the muscles, the skeletal muscle, smooth muscle, and cardiac muscle, all of them contain these two proteins in large amounts, actin and myosin. And you know that the myosin heads have to grab onto the actin and they pull. So that's shown here in this diagram. This is the myosin head, and that head kind of swivels and it has to attach to the so-called active sites of the actin, represented here with these blue balls. So they have to bind to the active sites. All right, so we don't want that binding when it's not time for the muscle to contract. And so there's this mechanism that keeps the myosin from accessing that actin active site. And that mechanism is a couple of other proteins. One of the proteins is called tropomyosin. It's the one that's shown here as the kind of this ribbon that wraps around the actin, and it acts as a chastity belt. If you know what a chastity belt is, if you don't, look it up. It's interesting history. But it covers the active sites. And well, we don't want it to cover it all the time either. So we have to make sure it stays in place, but then we have to make sure that it can roll out of the way when we need it to roll out of the way. And so the mechanism for this is another protein called troponin. And troponin is shown here. It looks like, I don't know, kind of purplish ovals, and it's attached every now and then along the tropomyosin. And it's like the lock that helps to keep the tropomyosin in place, covering up the actin active sites. So that's the lock. Well, every lock's got a key. The key in this case is calcium ions, and that is the secret behind calcium ions and the importance of calcium ions. Is these calcium ions come into the cell, they bind to the troponin and unlock the chastity belt. That whole thing kind of rolls out of the way. And then finally, the myosin head is finally able to attach to the active site on the actin. And that head can grab a hold of the active site and pull. And that's how we get contraction. And so the entry of the calcium during the action potential, remember we talked about how the cell lets some sodium kind of leak in and then it gets depolarized by opening up calcium channels. This is the calcium, it's coming in, it's all the keys, so that we can take off these chastity belts and let the myosin grab a hold and pull. Binds the troponin. The binding to troponin shifts the tropomyosin out of the way, exposes the actin binding sites to the myosin heads. And the myosin head binds, swivels, and moves the strands relative to one another, basically making them overlap more, which contracts the cell. Because the cardiac muscle cells are kind of coupled, they've got those gap junctions in them, they all kind of get the message at the same time. A bunch of calcium goes flooding into all of these cells, and contraction happens almost simultaneously. The contraction obviously squeezes the chambers. That's going to push the blood out, leads to emptying of the chambers. All of this costs something. It's none of this happens for free. So the myosin activity requires ATP to pull that myosin head back and then get it to swivel. That all, you have to pay for it using ATP. And then on top of that, all those ion pumps, the calcium pumps, the sodium pumps, the potassium pumps, they all have to be working to reset the ion levels back to normal before we can do all of this again. And so cardiac muscle is very energetically demanding. It needs a lot of ATP, which means it needs a lot of oxygen in order to do the cellular respiration, the aerobic cellular respiration, to get all this ATP. Yeah, it's a very oxygen hungry organ. This stuff's all covered on page 347. This diagram, and a part of the diagram that was on the previous page, is actually from page 98 in our book. This is from chapter 4 when we did the cellular biology. So this is repeating this, the myosin and actin cross bridge forming and contracting. That's going to require the presence of these calcium ions, and it's going to require the presence of ATP. And these filaments are passing over each other, sliding past each other. That's how the sarcomere works. Sarcomere is the organelle in which these proteins are part of. All right, so the calcium binds the troponin, moves the tropomyosin out of the way, the actin head, the myosin head attaches to the actin, and contraction can happen. And then we relax. The calcium ions get either pushed out of the cell or put away in the sarcoplasmic reticulum, an organelle inside the cell. They can store the calcium for us, and that's how we get relaxation. The myosin heads come off, and the fibers slide past each other again to the resting position. And then we do it all again. Right, the excitation and contraction coupling in the heart cells. So remember, we're going to do an action potential. That is, we're going to change the polarity on the membrane. That causes these calcium ion channels to open. A bunch of calcium comes flooding into the cell. That calcium is needed, as we just went over, for the uncovering of the actin, so the myosin can attach. And so the contraction keeps going on as long as calcium's available. Then when the cell repolarizes, remember we change, some ions have to move out of the cell, the potassium ions have to move out of the cell. When that's happening, we put away the calcium. So the inside the cell calcium levels, those drop. And we do it in a couple of ways. We push the calcium out of the cell, and we can store some of the calcium inside the cell in this sarcoplasmic reticulum, this organelle. It's basically the smooth ER. And so we have these calcium pumps, the calcium ion ATPase. It's a pump that pumps the calcium back into this organelle. And so it's not available to bind to troponin and make the actin available. All right, so the calcium gets sequestered, gets put away in this sarcoplasmic reticulum until the next action potential. And then we do another contraction. Now, previously, we talked about how the sympathetic nervous system can help the heart rate increase. And we talked about a bunch of different mechanisms that occur that allow the cells to depolarize faster. And you can get more depolarizations in a smaller period of time. Oh my goodness. Now we're going to talk about how the sympathetic nervous system can not just speed up the heart, it can make it pump harder. And the way it does that, it increases the contractile force. It increases how much calcium can come in. This diagram has a little bit more detail than I want to share with you, but I'll show you. So there's the beta-1 receptors that are on the outside of the cell, the norepinephrine from the sympathetic nervous system, and can stimulate that. There's some signaling that happens inside the cell. We talked previously when we were in the cancer chapter, we talked about kinases. Those are signaling molecules. Well, they're proteins inside the cell that phosphorylate something. Basically, this is all part of a signaling cascade inside the cell. I'm not going to worry about the details here. What I want you to see, though, is we're going to get increased activity at this calcium ion channel. So bringing calcium in from outside, it's coming into the cell. So more keys for those chastity belts. And we're going to store more calcium inside the cell in this sarcoplasmic reticulum. So next time we contract, we already have a bunch of calcium inside the cell. All we have to do is let it out, and we can still bring in more calcium from outside the cell. And so with all those keys around, that troponin, that is unlocked, the tropomyosin rolls out of the way, and all the actin is available for myosin to contract, and so it can contract hard. All right, we will get back to this concept of contractility shortly. But let's talk a little bit about something called stroke volume. Buff background means we're changing topics. The terms preload and afterload, let's define what they are and then talk about the impact that they have on the stroke volume. So preload is how much are we loading up the ventricle? How full can we get that ventricle? So it's the amount of stretch and pressure at the end of diastole. In other words, when the heart when the ventricles are relaxed, how full can we get those ventricles? And it turns out that within normal limits, the more we can fill the ventricles, the more the ventricles can contract. The stroke volume is higher. If you need an analogy for this, when you go to blow up a balloon, you don't just take a normal breath and start there. You go, and then you try to blow up this balloon. So by increasing the filling, we can get a bigger stroke volume, we can get more out. Okay, then the other concept is afterload. And it's unfortunate because it sounds like it's just the same thing, but afterload's a little bit different. So afterload is basically the pressure the heart has to push against to get the aortic valve to open. Going back to our balloon analogy, where you took the big breath in, you preloaded big. The afterload is if you've ever tried to blow up a balloon that's very hard to blow up, there's this moment when you're pushing as hard as you can and nothing is happening. There's no change in volume. Everything's just staying put, but the pressure is increasing. And so that pressure that's pushing back, that's the afterload. That's what you have to push against. So basically, if the patient is hypertensive, the pressure that's in the aorta is pushing back against what the ventricle is trying to do. So in this case, the afterload is working against you. The more afterload you have, the smaller the stroke volume, the less blood can get out of the heart. So in simplest terms, preload helps stroke volume, afterload hinders stroke volume. All right, so then let's go back to contractility. That's the ability to generate force during the contraction, and it's going to depend on those two things. It's going to depend on the sympathetic nervous system stimulation, that's norepinephrine action on the beta-1 receptors, and it's going to depend on the degree of preload that we get. And I'll talk a little bit more about that in a moment. And steps covered on page 348. Yeah, here's the diagram. All right, you may have heard of this concept of preload related to Frank Starling law of the heart. That's the only time I'm going to mention it. Basically, it's this. The greater filling of the ventricles, it's going to stretch the sarcomeres. It's going to pull the actin and myosin to just the right position where they're going to have optimal length for force of contraction. And I want you to think about other muscles in your body, skeletal muscles. So you know that if you want to contract your biceps, if you already start with your elbow bent, that muscle, the actin and the myosin, they're largely overlapped already. So like you can't go very far with this. But if you start here, you're a lot stronger when the fibers are not as overlapped, but that you want a little bit of overlap, and then you can get the best contraction. So on the x-axis, we have length. At this short length, this is already contracted. This is empty ventricles. They're not very stretched at all. You can't get a lot of force. But with the full ventricle, things are stretched out more. That's over here. You're much longer, the thing's more stretched out, we're all full. Then when you looked at the contraction curve, oh my gosh, we can get so much contractility just because of that perfect positioning of the actin and the myosin. Now, beyond that point, yeah, it goes downhill. If you stretch this too far, and think about it, like you try to pick up groceries with a completely straight arm, you can't pull that up very well. But if you get a little bit of a bend in your elbow, then you can pick that up pretty easily. All right, so there's this like optimal stretch that you want in the muscle, and that's what's achieved when we get that little bit of oomph from the atria pushing a little bit more blood into the ventricles. We get that little bit more stretch, and that allows us to get the most oomph, the most contractility. And that's on page 350. All right, so the greater filling, the greater preload produces greater stroke volume, and that's shown here. Let's just look at this purple line in the middle. The greater the volume, the greater the pressure is how they put it here, but it's related to the amount of stretch. So the more stretch we have, the bigger our stroke volume is within normal limits, right? If you put on top of that, add to that like sympathetic nervous system stimulation, the norepinephrine stimulation, you're going to get even more contraction out of this heart. On the other hand, looking at the red line, if we have a patient that's in heart failure, Yeah, getting a little bit more stretch may help a little bit, but oh my gosh, somebody with heart failure, even if you're stretching a lot, you're not even going to be able to do the output that you would get even at half full with a normally functioning heart. So that heart failure is going to significantly decrease contractility. The cardiac cycle, putting it all together. All right, so it's this recurring sequence of cardiac electrical activity and pumping activities. And we kind of try to tap into this. We detect things about the heart by taking pulses, different parts of the body. We listen to heart sounds, and we eavesdrop on the electrical activity using EKGs. The electrical events come just before the mechanical events. I mean, barely, but they come before. So for example, let's start up here. We have the P wave. That's the depolarization of the atria, the sinoatrial node and the atria. And so that gets picked up. And then all those atrial cells that depolarize, they're letting the calcium in. Calcium is the key to unlock the actin so that the myosin can hold on and we can finally get a contraction. Then we pass that message along the AV node, pass that to the ventricular cells, and then the ventricular cells are going to depolarize. That's the QRS complex. What's happening there? Hey, it's a bunch of calcium coming into the cells to unlock the troponin, move the tropomyosin out of the way so the actin and the myosin can bind to each other and contract. So then these ventricles push really hard. But there's that moment, that isovolumetric contraction, that's us blowing up the balloon. We're pushing real hard, but the balloon hasn't inflated yet. That's that moment. Then as systole continues, we finally are able to overcome the afterload, the pressure that's pushing back, and we can push the blood out into the aorta. Then those cells, the ventricular cells, they have to relax. When they relax, they can only relax when we repolarize. Those movement of those ions, that's what we're picking up on with the T wave. And so then we have relaxation. This is diastole. And that's when the ventricles can fill again. And then we start that whole cycle again and again and again. And that's the cardiac cycle. And so to summarize, the heart is a pump. It's going to put some stuff out. That cardiac output is the product of the heart rate and the stroke volume. How much cardiac output we need at any given time is going to depend on what the body's doing. How much oxygen do our tissues need right now? When we are at rest, we don't need as much, so the cardiac output chills. So the parasympathetic system slows the heart rate. The heart's not contracting as hard, so we're not putting out, so the heart rate is slower and the stroke volume is lower. Then with exercise, our oxygen demands increase because our ATP demands increase. So we better increase that heart rate and we better increase that stroke volume. And so we've already said the heart rate is under autonomic control. Parasympathetic slows it down, sympathetic makes it go faster. And then the stroke volume is also controlled partly by the autonomic nervous system. As we said, the sympathetic part of the nervous system can increase contractility. And remember, the way it does that is by allowing more calcium in and storing more calcium. But the other things that also play a role on stroke volume are how much preload did we get? by stretching a little bit to that optimal length, that increases the force of contraction. And then what's the afterload that we're dealing with? How much pressure is pushing back? And that's going to decrease how much we can eject from the heart. And that is the summary of the heart pump function and the end of part 3 of this lecture. See you in part 4.